In our previous videos, we have understood the control and coordination in human body. We also have to understand control mechanisms in plants. Do plants have control systems? Plants show response to external stimuli. The changes in the environment to which plants respond and react are called stimuli. The external stimulus may be light, gravity, touch, chemicals, etc. Since plants are anchored by their roots, they cannot move in response to environmental stimuli. Instead, they change their growth pattern. The changes in growth pattern in response to a stimulus may be by tropism, nastic movements, or by growth regulators called plant hormones or phytohormones. Plants respond to various stimuli very slowly due to the absence of nervous system. So in most cases, the response to stimulus cannot be observed immediately. Let us understand the types of change in growth pattern in detail. Charles Darwin and his son Francis described two broad categories of plant movements. The first one is tropism, which is directed growth in response to stimuli, and nastic movement, which is direction independent movement relative to the source of stimulus. Let us first understand tropic movements in plants. Tropism is a biological phenomenon indicating growth or turning movement of a plant in response to an environmental stimulus. This response is dependent on the direction of the stimulus. Tropism may be either positive, which is towards the stimulus, or negative, that is away from the stimulus. The different types of tropic movements are phototropism, geotropism, chemotropism, hydrotropism, and thigmotropism. What is phototropism? It is the growth response of a plant in response to light. Stems exhibit positive phototropism while roots exhibit negative phototropism. Geotropism. It is the growth of a plant part in response to gravity. Roots exhibit positive geotropism while stems and leaves exhibit negative geotropism. Chemotropism is the growth response of a plant to a particular chemical. Roots grow towards useful minerals in the soil but away from harmful ones. Another example of chemotropism is seen in plant fertilization and pollen tube elongation in angiosperms. Pollen, which contains the male gametophyte, is transferred to another plant via pollinating agents. If it is compatible, it will germinate and begin to grow. The ovary of the plant releases chemicals that stimulate a positive chemotropic response from the developing pollen tube. In response, the tube develops a defined tip growth area that promotes directional growth and elongation of the pollen tube. The next one is hydrotropism. The growth response of a plant to water is called hydrotropism. Roots exhibit positive hydrotropism. Thigmotropism. Growth response of a plant to physical contact or touch is called thigmotropism. Plants that cling to physical structures such as walls or fences exhibit positive thigmotropism. Let us look at one example naturally. Let me take you into my garden. Just observe this grapevine growing in my garden. It is a climber which naturally grows on trees. Here my grapevine has taken the support of the neem tree and it has grown towards the wall. You can observe slender structures called tendrils which appear on the top and sides of the stems. Tendrils are required to coil around the fence to reach for the sun and the heat. This kind of directional growth, when the plant part touches a solid support, is called thigmotropism. In climbing plants, thigmotropism helps them direct the pattern of growth around an object that is in contact with the plant. The next type of change in growth pattern is the Nastic movement in plants. Nastic movements are growth independent movements. The movement of a plant part in response to an external stimulus, where the direction of response is not determined by the direction of stimulus, is known as Nastic movement. It is not a directional movement. That is, from whichever direction the stimulus is applied, it affects all part of the organ of a plant equally, and they always move in the same direction. This movement takes place in parts like leaves, petals of flowers, etc. The types of nastic movements are seismonastic movement, photonasty, thermonasty, nyctinasty, and thigmonasty. Seismonastic movement. This type of movement is caused by shock, touch, fast moving wind, or raindrops. Photonastic movement is the non-directional movement of a plant part, usually in the petals of flowers in response to light. Thermonastic movement. Movement in plants which occur due to change in temperature. It is thermonasty. Movements caused due to variations in light and temperature are called nyctinastic movements. It is also called sleeping movement. The non-directional movement of a plant in response to the contact of an object is called thigmonasty. Have a look at another plant here. The moment I touch the leaves, they fold. This is the Mimosa pudica, which is called Attipatti in Telugu and touch me not plant in English. This plant belongs to the Fabaceae family. Mimosa pudica shows nastic movements. They are non-directional responses to stimuli. They differ from tropic movements. There are many types of nastic movements. Mimosa shows thigmonasty, which is the response to touch. What happens when we touch the leaves of this plant? Mimosa pudica is a plant with compound leaves which droop when they are stimulated. When we touch the leaves, an action potential, that is, an electrical impulse, is generated. At the base of the leaves, there are pad-like swellings called pulvini. Pulvini helps to hold the leaf erect. Here the cells contain lot of water, and water pressure pushes against the cell wall, creating what is called turgor pressure. So the cell is rigid, and thereby the leaf is also erect. When the leaves of mimosa are touched, there is a change in the turgor pressure at the pulvinus because of the action potential, and water is channeled to the other side of the leaf. Pulvinus loses its firmness, and hence the leaves fold. Plants do not have a well-defined endocrine system, yet they can sense the presence of light, heat, touch, etc. Their mechanism of control is by means of chemicals or hormones. Hormones present in plants are called phytohormones. These phytohormones control one or the other aspect of growth of the plant. Therefore, plant hormones are also called growth substances. Let us look at some of the major plant hormones and their action. Auxins. This plant hormone helps in cell elongation, which is increase in the size of the plant cell. It also helps in differentiation of shoots and roots. Differentiation means it is a process where cells of shoots and roots mature to perform specific functions. The next plant hormone is cytokinin. They promote cell division. A lateral bud is a meristematic area on the side of a stem that gives rise to side branches. These lateral buds are inhibited by auxins. Because auxins move from apical meristem, which is the bud at the stem's tip. This phenomenon is called apical dominance. If the apical meristem is removed, oxygen concentration drops. Cytokinin is promoted, causing the lateral bud to form. Thereby, cytokinins promote sprouting of lateral buds. They delay aging in leaves. Cytokinins also help in the opening of the stomata. The stomata have guard cells. Cytokinins induce the influx of potassium into the guard cells so that they remain turgid and they open. The next hormone is gibberellin. Gibberellins help in germination of seeds and sprouting of buds. Germination of seed means it is the awakening of the embryo at the end of dormancy, giving rise to seedling. It is the process of seed developing into new plants. The next hormone is abscisic acid. This hormone helps in closing of stomata. It stimulates the loss of potassium ions from the cell and decrease in turgor pressure. Therefore, the stomata close. Abscisic acid also helps in seed dormancy. Seed dormancy is an evolutionary adaptation which will not allow seeds to germinate in unsuitable ecological conditions. Thereby, it leads to delayed germination. Abscisic acid maintains seed dormancy. It promotes aging of leaves. Aging in plants is also known as senescence. Leaf senescence is influenced by abscisic acid. Thus, it helps in recycling of nutrients and growing new leaves. The last hormone we will be learning is the ethylene. Ethylene helps in fruit ripening. It helps to change color, texture, aroma and flavor of the fruit. It makes the fruit more palpable. Let us try to understand some early experiments in phototropism. Early experiments in phototropism were conducted by Charles Darwin and his son. He noticed that if light is shone on a coleoptile, which is a shoot tip, from one side the shoot bends or grows towards the light. This bending did not occur in the tip itself but in the elongating part just below it. Removing the tip or covering it with foil meant that the shoot would no longer bend towards the light. Covering the elongating part of the shoot did not affect the response to light at all. So Darwin concluded that some influence is transmitted from the tip to the more basal regions of the shoot, thereby regulating growth and inducing curvature. Boyce and Jensen continued investigating these responses. He cut the tips of coleoptile and placed a thin piece of silver between the coleoptile and the lower shoot, between the tip and the lower shoot. The result was that the shoot did not grow or curve towards light. When he repeated the experiment using a block of agar, the result was that the shoot grew and curved towards light. Thus, he concluded that the Darwin influence was a water-soluble chemical which is capable of fusion through the agar to the lower elongating part of the shoot where it had its effect. This water soluble influence was later identified as the plant hormone auxin. A third scientist went, cut the tips of coleoptile and placed them in the dark on agar blocks. He thought the agar would absorb the growth promoting chemical. He then placed the agar blocks on the tipless coleoptiles. When agar block was centered on the top, the coleoptile grew straight. If the agar block was offset, the chemical would have uneven distribution and the shoot would curve. This proved that the response was due to a water-soluble chemical that diffuses from the tip of the plant down to the side of the coleoptile, causing it to curve towards the light.